this is also another important statement that I have borrowed from someone, that subject is made by the person. Person doesn't make the subject. Okay. So now the choice already you have made, and here actually this drug discovery and development, it is something where we have a role, and it identifies and validates drug receptors, drug targets. So drug development has changed. Nowadays we don't do it by the older method. We have all this computer aided design, the thorough output systems. So it has been digitalized, and we would like to see the pharmacokinetic profiles like the absorption, distribution, metabolism, excretion, and we assess the drug receptor interactions and dose response relationships. So preclinical studies in Assam, in Jorhat, NEIST, agota RRL boli koise, Regional Research Laboratory. So most of the preclinical studies we get the data from Jorhat. Okay, they have a very good animal house facility and they are expert, and we take the help from them. Then the drug has to be safe first. So we need some safety testings. So we use this acute, subchronic and chronic toxicity testings, and these testings are normally done in animal models. So identifies potential adverse effects early in development and helps determine the therapeutic index. I hope all of you know what is therapeutic index. Therapeutic index is the safety margin of a drug. It is LD50 by ED50, median lethal dose by median effective dose. And these are all statistical calculations. You need to know that a drug has to be minimum, therapeutic index should be more than 1. Okay. Clinical trial support. So the first in-human studies and dose escalation protocols. Now, whenever we have thought that okay, this drug is safe now this human studies and what should be the starting dose, because you just cannot go on a very high dose. So dose escalation protocols are there, and we need to observe all these things, and we interpret the pharmacological data from different clinical trials. Now please understand that all these clinical trials are undertaken considering some fixed parameters. Now when we develop a drug, the patients, they are kept in ideal conditions. So many drugs, when they are marketed in the field, they may behave abnormally. So those things we need to consider. So we collaborate with clinical teams to monitor efficacy and the effects. Now this is something which I actually want my visitors to put some effort in attending. This is a very novel science. It's pharmacometrics. Okay. Now this quantifies the interaction between drug and patients by interlinking biology, physiology, and pharmacology with disease condition through some mathematical tools. The data and information collected from different sources are linked together quantitatively. The quantitative answers are required for the questions raised during clinical use regarding safety, efficacy, and dosage regime, and more specifically to design individualized dosage regime. It's a model called as non-linear mixed effect models, and which has been very popular. Especially, it's a gold standard by US FDA, and this model is specifically used for testing drugs in infants or in pregnancy, because ideally it is very difficult to test drugs on pregnant ladies and infants. So we use these kind of models, and recently actually this is one paper that I published, not published actually, on the verge of publishing, improving data integrity